Thank you all for coming today, and I'm really excited to tell you about this, actually several stories in both melanoma and another very important cancer to me, and it's that cancer that provides us the history for what I hope to be is a major success story that is up and coming in melanoma. So I'm going to talk first a little bit about stage 4 melanoma and what the current treatments are. Go over how we get new drugs from a basic science laboratory into patients. Give you, again, a history of how targeted therapy evolved and show you what the potential impact of this approach is when it's done well. I'm going to introduce to you what I think are the main targets in melanoma. Go over briefly the current clinical trials we have at the Huntsman that approach this unique paradigm. And then finally, I think it is only through combination approaches that we will achieve a lasting cure in melanoma. So first off, stage 4 melanoma. Dr. Leachman presented this a little bit earlier, but basically that is melanoma that has escaped the site of origin. And it's spread beyond its normal lymph node drainage basin, and it can involve really 3 different classes of sites. One would be distant skin, subcutaneous nodules, or lymph node metastases, and those are lymph node metastases, again, that are outside the normal drainage pattern. Lung metastases would involve M1b disease, and then finally M1c is all other sites. And one of the extraordinarily frustrating things about melanoma is this is a cancer that can go so many places. Obviously, having, as you march down this list here, it gets from relatively easy to treat to extremely difficult to treat. So what really are the goals of treatment in stage 4 disease? Well, first off, as with any cancer, we have to ask the question, can we cure it? And I think there are unique melanoma patients with stage 4 cancer that can achieve what I would consider maybe not a cure, but a durable, complete remission from their cancer. Can we prolong life? I think the answer is definitely yes for some patients. And then finally, can we delay the onset of symptoms related to cancer and by doing this improve quality of life? And I would say definitely yes, we can do that for just about everybody with stage 4 cancer. So currently we have, I would say, 3 broad categories of therapy that have been used over the years in stage 4 melanoma. The first one on my list is high-dose interleukin-2. This is still the only FDA-approved therapy that has produced durable, complete responses in stage 4 patients. And even though you'll hear about some exciting new frontiers, I think that this therapy still has significant importance in the treatment of stage 4 patients. Also, biochemotherapy, again, has been a therapy that has shown durable responses, and I think for select patients this is a very important treatment option. And I think it will remain a part of our treatment armamentarium for some years to come. And then finally, the standard approach for many in the community is this drug called the carbazine, and it would fall into a category of more classic chemotherapy. It works primarily by taking advantage of the fact that cancer cells divide fast, and that by damaging their DNA, we can put the brakes on them with this type of drug. The things that are on the horizon that I think are particularly excited, first off, immune stimulators and a drug called ipilimumab. You'll hear a great deal about this drug from Dr. Kirkwood here in a moment. And then targeted therapy. And the 2 targets I'm going to talk about today in some detail are c-KIT and BRAF. These are very important in the development and progression of melanoma. But there are many targets in melanoma that are not on this slide. So I just want to introduce you to the concept that melanoma is relatively complex. So to get a new drug from the science lab to a phase 3 clinical trial and ultimately an approval by the FDA, it takes a lot of steps. First off, we have to understand something about the cancer that we're trying to treat. And what is becoming increasingly important is to pick apart exactly what the target is within a cancer cell that we want to hit. We then design drugs and synthesize them to hit that specific target. And then once we've got something that looks pretty good, we try and make it look even better by tweaking that molecule and making it stronger and more specific. We then have to get it formulated so that it actually can get into the body and get to the site of a tissue. We take great care to make sure to pilot that in animal models and get a very good understanding about how this drug is going to work before it goes into humans. And then it has to be scalable. We have to make enough of this drug in a cost-effective way to make it available to all the patients who need it. And once you're up to step 6 here, that's when you make a proposal to the FDA to suggest that your drug is ready for treating humans. And in humans, we have to go through a series of trials. First off, phase 1, and the purpose of a phase 1, number 1 is to hopefully pair a phase 1 with just the right patient so that that patient participating gets some benefit. But also importantly is defining exactly what the toxicity of our drug is. Is it safe? Where does it go in the body? How does it clear? These are all of the questions that we try and answer with the phase 1 clinical trial. The hope is that by doing that, we get some signal in a small group of patients, and then we can expand that to more patients and see how many out of a larger population actually get benefit from our drug. And that's the purpose of a phase 2 trial. If that goes very well, then we have to compare this new drug to what's the community standard and prove that it actually is better than what we're already doing, and that's the purposes of a phase 3. And I would say all along the way, at each of these steps, as we expand to more and more patients, we really have our eyes on the toxicity and the safety of this drug, and those are really important when we're considering any clinical study. So I know you're here today to hear about melanoma, but what I want to tell you about is a story of how targeted therapy came to be and why it's so important in all cancers and why it's important in melanoma. And this really involved in a cancer called CML, or chronic myelogenous leukemia. The important things to note about the success story is it was really a collaboration of companies and physicians and scientists in academic institutions like the Huntsman. Two of the main players here, Nick Leiden was working at a company which now is called Novartis, and Brian Drucker, who is a mentor to me. As a graduate student in Oregon, he was on my thesis advisory committee, and really this story is the inspiration for why I chose to do cancer and for what I hope happens in melanoma over the next several years. And what these folks did is they first identified a target. They developed the tools to test their drugs in a test tube to really define them before they ever went into a patient. And then they tested their drugs in exactly the right patients. And they also pushed through some obstacles to drug development, and particularly they had chosen a relatively rare cancer here, and that presents some challenges. And just to illustrate how rare this cancer is, it's uncommon. It's only about 1/10 as common as melanoma. It is a cancer of the blood cells, so for that reason it is relatively different than melanoma. But what made it the cancer to test this concept in is that there really is one target in CML. There is one switch. And so what they set out to do is to find that on switch and turn it off. And this is the on switch in CML, and it actually involves a swapping of DNA between 2 different chromosomes. It takes this gene, called the Abelson tyrosine kinase, and puts it in juxtaposition with this other gene called BCR. And after that happens, several things occur within the cells. They divide too rapidly, they no longer know to stay in the right place, and they lose the ability to recognize that something's wrong with them. Most of the cells in our body actually have a self-destruct mechanism, and when they start marching on their way towards cancer, they die. This pathway parallels exactly what happens in melanoma. It just happens to be much more simple in CML. So how do we turn that switch off? Well, quite simply, it involves a lot of work. The chemists and the physicians had to go through screening thousands of chemicals to see exactly which one would sit at their target and block its activity. And that target, again, was called a kinase. After they answered the question, can we turn it off, and they found one of these many molecules that could actually work, then they had to set about tweaking that molecule to make it more specific. Just to give you a little scope of the problem that they were approaching, this is a a family tree of all of the potential targets that are kinases within our human bodies. We have about 518 of these kinases. Up here, sitting at this black arrow, is the Abelson kinase. And so that was the single target that they were really after in developing their drugs. The green arrows, which I don't think project very well, are several of the targets of which I think are important in melanoma. And I'll get to talking about those here in a little bit, but I want to at least follow this story through its conclusion. So this is actually represented in white, a picture of the drug that came out of all of that screening. All those thousands of molecules, this is the one that seemed to sort out and inhibit their Onswitch. So good chemists are kind of like kids that play with Legos. And chemistry is really like putting together new Legos to make the big Lego do something slightly different. And that's exactly what they did. They first added these different groups here to make the drug more active and more specific for their target. But I think the most important addition was this group over here. And this is what allowed this drug to be taken as a pill. It allowed it to be relatively non-toxic. And it made it just so much more convenient for patients. This is a picture of the distribution of activity of that particular drug. This is Gleevec or imatinib. And I'm hopeful that those in the back of the room can appreciate that this little tree here is actually shrunk down from what I showed you earlier. All of these red dots are sitting down on different parts of that tree. And this, again, the black arrow, that represents the Abelson kinase. Since the discovery of Gleevec, there have been maybe as many as 100 different types of molecules that are looking at hitting different parts of this tree. And you can see some of them take out the whole tree, and some of them take out just a small limb, like imatinib. So just to give you some sense of timeline and how long this took to actually develop, in 1996, this was about my second year of graduate school, is when Dr. Drucker was at this phase of the development of the drug. They were just about ready to go to the FDA and ultimately file to allow human studies. And to make a long story short, this was one of the most rapidly approved drugs ever by the FDA because it worked so well. And I'll show you the slides to illustrate that. So in 2001, just a short time after it was really approved for use in humans, it had its approval in second line. The impact of this was extraordinarily huge. Again, I told you that this is a relatively rare cancer in comparison to melanoma. But over the years, and this is looking at about a 5 to 6 year time horizon, the patients that have been receiving Gleevec have survived much longer than those that had received the traditional drugs such as interferon and chemotherapy. And if you actually measure the number of people that were benefited by this in this rare cancer, there may be as many as 200,000 people alive with CML in the United States as a result of the discovery of this drug. So the potential impact for pairing exactly the molecular switch to the correct drug is absolutely huge. And if this were to happen in melanoma, it would be equally big, if not bigger. Now, of course, honors come to the people that develop this kind of work. They get great satisfaction, of course, in the lives saved. But also, they go on and win prizes. And probably one of the greatest prizes Dr. Drucker received was he got to carry the torch for the Salt Lake Olympics in 2002. And he's shown pictured here. This is a midday afternoon picture in Portland, Oregon, where I'm from. Sometimes it's not very sunny there, but anyway, Brian is somebody very special to me. And again, it's this story and him that inspired me really to work day and night to try and do this in melanoma. So how do we do that? What are the targets in melanoma that we need to go after? Well, we actually know quite a bit about how melanoma works, and this is group work that's been carried out by a variety of investigators, including Boris Bastian, who's not listed on this slide. But the pathways of how melanoma gets turned on really hinge through two arms, one of them going through a very important molecule here called BRAF, going over here towards proliferation, and another one going through a molecule here called AKT, causing survival. Up here are the receptor tyrosine kinases. And just like that Abelson tyrosine kinase I told you about earlier, there's a molecule in melanoma called c-KIT, which behaves very similarly. So one thing you might ask is, what if we took that great drug in this blood cancer and we tried treating melanoma patients with it? What would happen? Well, here is that c-KIT oncogene that I told you about, and it actually happens to be a very good target for Gleevec, or the drug that I told you about in CML. And here it is pictured just a little bit differently. So here is the CML target. Here is the melanoma target for that particular molecule. And early work done showed the potential of this. So this is a patient that had a very dramatic response when she took Gleevec. And within about 4 weeks, the activity of her tumors, which is in kind of red hot here, quieted down very significantly, and she had a near complete response. I don't know the long-term follow-up of this particular patient, but there have been others like her that have had relatively durable responses that have gone on for months and months. So the potential of this is obviously really important. Probably the largest group of patients presented to date actually is from a group in China, and this was presented at this last year's ASCO meeting in June. There they screened 390 patients, and they found 67 of those that had a KIT mutation or amplification, meaning they'd be great targets or great candidates for this particular drug. 21% of those patients that have been treated so far have had partial responses, and 35% have had stabilization disease of 4 to 10 months. Now, this is not a home run, but it's certainly a base hit, and these numbers are very comparable to some of the other treatments that we've had available in melanoma. So this concept clearly works. The challenge with this is c-KIT is not the most common kinase mutation in melanoma. And in fact, in cutaneous melanoma, which is far and away the most common melanoma, it only represents a very small portion of the pie chart over here. The big player is BRAF. And the good news is that we now have a couple of very good drugs that target that particular kinase. And this is where BRAF sits on that family tree of kinases, right over here in this branch. And to fast forward through a lot of work, this data reflects the work of hundreds of scientists, clinicians, nurses, nurse practitioners. If you remember that list from the beginning that Dr. Leachman shows, all of those types of people were involved in this work. And this was presented at last year's ASCO by Dr. Flaherty. To orient you to what this graph means, this is a percent of either growth on the top or shrinkage on the bottom. And each little bar here represents a patient along this line. Every patient below this line enjoyed shrinkage of their tumor, and some patients had very dramatic shrinkages of their tumor. This one here, having all of their cancer go away. That's really exciting in stage 4 melanoma. I'll take your question at the end. So based on these findings, we've really tried to craft our clinical trials focus at the Huntsman Cancer Institute to very carefully select patients for appropriate therapy based on their mutation type. And so we're testing all of our stage 4 patients for these mutations, c-KIT, BRAF, and NRAS. And for those that have a c-KIT mutation, we try to pair them with a kinase that has a profile that hits not only c-KIT, but several other very important targets in melanoma. For those that have BRAF mutations, we have 2 studies, both this study using a drug called Plexicon, which is the one I showed you in the last picture, but also a GlaxoSmithKline's BRAF inhibitor. And then finally, for patients that have an NRAS mutation, we're still not quite sure exactly how to target this, but we have some very good ideas. So we're pairing patients with NRAS mutations to appropriate phase 1 studies. Because the focus of my talk has been targeted therapy, I don't want to leave you with the impression that immunotherapy is not important. In fact, it is every bit, or if not even more important than the targeted drugs that I've told you about. Immunotherapy has been the regimen that has really provided the lasting, durable responses in melanoma, and it's for that reason that I think if we're to succeed, it has to be paired. And then finally, biochemotherapy is equally important because of its traditionally high response rate and those durable responses. So how do we cure melanoma in stage 4 patients? Well, I think going forward, as I said just a moment ago, we have to pair targets with immunotherapy. And we also have to combine targeted drugs. As I showed you, there's that tree of 518 different kinases. That's actually the tip of the iceberg. It may sound like a lot, but now we have tools that are exponentially better than Dr. Drucker had back in 1995 when he began his work with Gleevec. So I think that all of that will just serve to accelerate our progress forward. And it really does take teams. And here we have a fantastic team of very dedicated people. And this is only a short list of the roles that they fill. So finally, I'd really like to thank the AIM at Melanoma Foundation for giving me the opportunity to speak, and Valerie Guild for organizing this. And then finally, all of these familiar faces that I'm seeing in the audience, I really appreciate you all coming today, both patients and families, for their support of this. Thanks.